In the previous video, we learned about the first two steps in cellular respiration. In the first step, glycolysis, the glucose is going to be broken down into two 3-carbon molecules called pyruvate. If there are adequate supply of oxygen, the pyruvate will then move into the matrix of the mitochondria where it will undergo pyruvate oxidation. The end product of pyruvate oxidation is something called acetyl-CoA. So now we are going to look at what happens to the acetyl-CoA in the next step of cellular respiration, which is the Krebs cycle. In Krebs cycle, the acetyl-CoA is going to be completely broken down into carbon dioxide. We saw that in the second step in pyruvate oxidation, the 3-carbon pyruvate, you actually remove the carbon dioxide from it already, right? So now we are down to a 2-carbon molecule. So we're gonna release 2 more carbon dioxide, and then it will be completely broken down. All the glucose, all the carbons in the glucose molecule would have been released in the form of carbon dioxide after it goes through the Krebs cycle. So to begin with the cycle, we actually will take the acetyl-CoA, which again is a 2-carbon molecule, and that's going to be combining with a pre-existing molecule in the matrix. So there is something called oxaloacetate, which is a 4-carbon molecule, and this 4-carbon molecule is already in the matrix. So when the acetyl-CoA comes in, it will combine with the oxaloacetate, which then generate a 6-carbon molecule known as citric acid. For this reason, because citric acid is the first product that's formed in the Krebs cycle, that's why the Krebs cycle is sometimes also known as the citric acid cycle. So the citric acid is going to undergo a series of steps where at the end of it, at the end of the cycle, you are going to be regenerating oxaloacetate. Okay, and that will allow the oxaloacetate to combine with another acetyl-CoA, and then the cycle keeps on spinning and spinning. It's probably easier to visualize it with a diagram like this. So here we have the acetyl-CoA, and again, the acetyl-CoA, it's from step number 2, right? From pyruvate oxidation. Okay, and just to reiterate, everything that I'm talking about here actually happens in the matrix of the mitochondria. So the acetyl-CoA will now combine with the pre-existing 4-carbon molecule, right? So this is 2-carbon. Here is the 4-carbon molecule called the oxaloacetate. And once they come together, then you will have this 6-carbon citric acid. Okay, citric acid. So the citric acid is going to undergo many steps, which we're not going to go into. And in the process, what's going to happen is the citric acid is going to be releasing 2 carbon dioxide molecule. So citric acid has 6 carbon atom in it, and after you release 2 carbon dioxide, 6 minus 2 will bring you back to 4 carbon. Okay, so that's how you regenerate the oxaloacetate, and then it's just going to combine with another acetyl-CoA that's coming in from the pyruvate oxidation. Okay, there is going to be a video link to an animation that allows you to see this in motion. You will probably have a better understanding after watching that. In addition, the Krebs cycle is also going to be producing 1 ATP per spin, and then it's going to be releasing quite a lot of high energy electrons. So those electrons are going to be captured by 3 NAD+, creating 3 NADH, and then there is also the other energy carrier, the FAD, and it's also going to be picking up some electrons, and it's going to be generating FADH2. So just to refresh your memory, both NAD+ and FAD are energy carrier. They will be capturing electrons, high energy electrons, and taking them to the ETC, to the electron transport chain, to generate ATP. The difference between NAD+ and FAD is that NAD+ is able to sort of capture more energy compared to FAD. So later on you will see each NADH molecule will be able to create more ATP compared to each FADH2. Okay, so for every glucose that you are going to metabolize, the cycle will spin twice. Okay, that's because one glucose will be split into two pyruvate, right, in glycolysis, and then these two pyruvate will each undergo pyruvate oxidation, creating two acetyl-CoA. Okay, so from the two acetyl-CoA, they will each combine with a separate oxaloacetate, and so this cycle would be happening 2 times for every glucose that you break up, which means all these things, all these products that you're going to be creating are going to be doubled per glucose metabolized. So here is a summary of what I just described to you. I'm not going to read it to you again, but please do go over it yourself. I do want to mention this point though. I told you FADH2 carries less energy compared to NADH. So to be specific, each FADH2 will only have enough energy to produce 2 ATP molecules. Okay, so these ATPs are going to be created later on, right, in the electron transport chain. Right now, they're just kind of carrying these electrons to the ETC, where they will be used to make the ATP. So when you do that in step 4, the energy carried by the FADH2 can only create 2 ATP, whereas each NADH will be able to produce a maximum of 3 ATP molecules. If I just go back to the previous diagram, previous slide, you can see ATPs are generated in the citric acid cycle, in the Krebs cycle. So this ATP is going to be generated by substrate level phosphorylation, which is the same process that was used to generate the 4 ATP in glycolysis, right? So just to recap, in substrate level phosphorylation, there is going to be some kind of molecule with a phosphate, and it's willing to donate this to ADP. Okay, so an enzyme is going to come along, okay, and then it's going to bind to these two things, okay, and then it's going to help move the phosphate to the ADP, where it will then become ATP. Okay, so the 2 ATP that's produced in the Krebs cycle, right, per glucose, that's going to be created by substrate level phosphorylation. Okay, so I just want to clarify one more time. These energy carrier will be creating ATP later on in step 4. Okay, so we don't count those ATP until they get to step 4. So in Krebs cycles, these are the only ATPs that are going to be produced. Okay, and there will be 2 ATP molecules produced per glucose broken down. So here is a summary of what happens in the Krebs cycle. Okay, again, it takes place in the matrix, and the matrix once again is the inside of the mitochondria. Okay, it's in the fluid inside the inner membrane. So in this process, acetyl-CoA will be broken down into carbon dioxide, and for each glucose that you are metabolizing, the Krebs cycle alone will generate 4 CO2, 6 NADH, 2 FADH2, as well as 2 ATP molecules. So by now, glucose is completely broken down. You kind of create as much ATP as you could from glycolysis and from Krebs cycle. Much of the energy that was released through the oxidation of glucose has been captured by these energy molecules. Okay, so you have these NADH from each of the steps, and then you have some FADH2. Okay, so these are energy carriers that will take those high energy electrons that was released from glucose breakdown, and we are going to take these high energy electrons to the final step of cellular respiration, which is called the electron transport chain, or ETC. So that's pretty much what I just said, right? All the energy carrier will be transporting electrons to the ETC, and the ETC is located in the cristae of the mitochondria. So in case you forgot where the cristae is, the cristae is basically the finger-like projection, or these foldings that you have created by the inner membrane, right? And these foldings actually increase the surface area of the inner membrane, allowing you to pack more electron transport chain into them. So what exactly is the ETC? The electron transport chain is basically just a series of membrane proteins, and they are responsible for passing down these high energy electrons, and in the process they're going to harness their energy to create what is called a proton gradient, and I'm going to explain more in a diagram later. And that proton gradient will be used to generate ATP ultimately. So here is a simplified view of what's going on. You're going to have the electron transport chain, which are again membrane proteins. The NADH will be depositing the high energy electron at the top of the electron transport chain, and as the electron is being passed down the membrane proteins, the energy from the electrons are going to be harnessed to ultimately create ATP. And at the end of the electron transport chain, there is going to be an oxygen molecule that's going to be taking the electron away, so that you will form water as a byproduct. Okay, so if you remember the overall equation, glucose is going to be oxidized into carbon dioxide, whereas the oxygen is going to be reduced into water. Okay, so reduction, right? You might remember GER, right? Gain of electron is reduction. So when oxygen gains the electron, that's going to cause it to become water molecule. Okay, so this here is basically what the ETC looks like and how it works. I'm just gonna try to explain it on a drawing for you, and you can draw along with me, which might help you understand the process a little bit better. Okay, so let's take a look at how this ETC works. I'm gonna have the mitochondria here. That would be the outer mitochondrial membrane, and then here we are going to have the cristae, the inner, formed by the inner membrane, and of course on the inside, that's where the matrix is going to be. So we're gonna zoom in into this area here. Okay, so we are gonna draw that much bigger scale. So there we have the inner membrane, and they are folded up to increase surface area, so you can pack more electron transport chain in. Right, so this is the inner membrane. Okay, and these foldings are called cristae, and here, right, just for reference, that's gonna be your outer membrane. Okay, outer membrane. Okay, and over here, the space between the outer membrane and the inner membrane, that's going to be the intermembrane space. Intermembrane space. And the fluid that's inside the inner membrane, that is going to be the matrix. Okay, so the matrix is where the pyruvate oxidation occurred, and also where Krebs cycle occurred. Right. So now we're going to add some electron transport chain over here. Let's choose a nice color, and I'm gonna put the electron transport chain here. Okay, so for simplicity, I'm just gonna put one electron transport chain. Okay, so they are basically membrane proteins that are embedded within the inner membrane. Okay, so this whole thing here, that's going to be the ETC, and again, they are membrane proteins. So what's going to happen is we are going to have a high energy energy carrier, right? So we have the NADH as well as the FADH2. But first, let's focus on the NADH. Okay, so the NADH is going to come to the top of the ETC, right, to the first membrane protein in the ETC, and what's going to happen is the NADH will deposit its electron. Okay, so the two high energy electrons are going to attach to the first protein, and in doing so, you are going to regenerate NAD+. Okay, and of course H+. Okay, so what's going to happen is the NAD+ now will be able to go back to pyruvate oxidation as well as a Krebs cycle to pick up more electron, and then it's going to be able to come back and transport them to the ETC. Okay, so let's focus on these high energy electrons. Remember, they are deposited by the NADH. Okay, and so these electrons will be passed down the electron transport chain. Okay, it's like a game of hot potatoes. Okay, so this one will pass the electron to the next one, and then it will pass the electron to the next one. Okay, so going down a slide. As the electron is passed down the ETC, what's going to happen is the protein will harness the energy from this high energy electron and use that energy to pump H+ from the matrix into the intermembrane space. Okay, so we are going to pump H+ into the intermembrane space. Okay, so as electron goes down the ETC, the energy is used to pump H+. Okay, now at the bottom of the ETC, at the bottom of the electron transport chain, we are going to need a molecule to take the electron away. Okay, think of this as a slide. Okay, so one kid goes down the slide, like a parent has to take the kid away before the next kid can slide down the slide. Okay, if the electron is stuck at the bottom, then the next electron can go, right? So we must have something to take the electron away. Okay, and that something is going to be oxygen. Okay, so oxygen is going to be called the final electron acceptor. Okay, without the oxygen, then the electron will be stuck on the ETC, and you won't be able to do this any further. Okay, we must have oxygen to remove the electron from the bottom. Okay, so the oxygen will combine with the electrons, and then it's going to form water. Okay, so that's why oxygen is said to be reduced into water. Okay, it's reduced because it's gaining electron. Right, so this is gonna occur. We also have the FADH, right? FADH2. So FADH2, as I said earlier, is going to have less energy. Okay, so FADH2 will deposit the electron at a later point on the ETC. Okay, and then it will become FAD again. So the FAD can go back to the Krebs cycle and pick up more electron. Okay? So since the FADH2 deposit the electron at a lower point, it won't be able to pump as much H+ into the intermembrane space. Okay, so that's going to be important later on. Okay, so for now, what's going on is NADH2, FADH2 brought all the electrons from the first three steps, and then they are going to deposit them onto the ETC, and the energy is going to be used to pump H+ from the matrix into the intermembrane space. So as this process keeps on happening, over time you are going to have a lot of H+ in the intermembrane space. Now, the H+, they are positively charged, and as you learn in membrane transport lecture, charged particles cannot go through the cell membrane, right? So the inner membrane is similar to the cell membrane in that it's also made up of phospholipids bilayer. So these H+, they really want to come back in, but they can't, okay, because of the inner membrane, right? So over time, as more and more electrons are being deposited at the ETC, you will create a high concentration of H+ here. Okay, so there will be a high concentration of H+. Right, and on the inside here in the matrix, you are going to have a low concentration of H+. Okay, so there might be a few H+. Okay, remember you're generating these H+ every time the NADH is converted into NAD+, and anytime FADH2 is converted into FAD, right? So most of it will be pumped into the intermembrane space, but you will still have a few. So what's going to happen is the H+ really want to come back in, and so the only way they could go back into the matrix is through a special protein. Okay, a special protein. So the special protein is not really part of the ETC, okay, but it's going to be nearby. Okay, it's going to be close to the ETC. Just going to choose a color here. So there is that special membrane protein. Okay, they actually do look like that. It looks like a doorknob, and this is called the ATPase. Okay, ATPase, or sometimes called ATP synthase. Okay, so same thing. Okay, so this will allow the H plus to come back in to the matrix. Okay, so these H plus will now go through the the ATPase. Okay, and what's going to happen is the ATPase will harness the energy from the gradient. Okay, so as the H plus comes in, they will lose energy. Right, this is kind of like building a dam, right? So you have all the water stored up here, and they want to go downhill, and then you put a turbine in there, right? So as the H plus goes down the concentration gradient, they will lose energy, and the ATPase is going to be able to take that energy and use it to create ATP. Okay, so ADP plus P phosphate, right? And with the energy from the H plus going down, these will combine and it will form ATP. Okay, so how many ATPs are you going to be able to create? Well, if you're looking at the NADH, then it is going to be able to pump enough H plus to create 3 ATP. Whereas the FADH2, because they deposit the electron at a later point, they will only be able to pump enough H plus to create 2 ATP. Okay, so 3 ATP per NADH, and then 2 ATP per FADH2. Okay, so to create ATP through this method, it is called oxidative phosphorylation. Oxidative phosphorylation, as opposed to substrate level phosphorylation, right? In glycolysis and Krebs cycle. Right? So this phosphorylation, the addition of P onto ADP, uses oxidative reaction, okay, to power itself. So that's basically what all the events that takes place in the electron transport chain. So here is the better drawing, right? The more professional one. You can see the electron transport chain, the 3 proteins. You have the NADH depositing the electrons, and then it's going to be passed down the chain, and the energy is used to pump H plus. Okay. You would have FADH coming in at a later point, recreating FAD, and then that's also going to be pumping H plus, and all these H plus want to come back in, but they could only do so through the ATP synthase, and as they come back down, the energy from the H plus going down its gradient will be used to add phosphate to ADP, creating ATP. In order for the chain to continue passing down electrons, you must remove the electrons at the very end, and that's the job of oxygen, right? Acting as the final electron acceptor. So here is a little skull icon that represent hydrogen cyanide. Okay, so as you might know, cyanide is very poisonous, and the reason is it will basically bind to the ETC and prevents the electron from getting off, okay, because the CN cyanide is very electronegative, okay, compared to oxygen. That means it's going to have a high affinity attraction to electron, more so than oxygen. So in the presence of CN, the electron will not want to get off the ETC, and it will just remain there. So it clogs up the whole ETC. When the ETC is clogged up, then NADH will not be able to deposit its electron because it's already full, right, the chain. So if you can't deposit the electron, then you cannot generate additional ATP, and that's why the cyanide is going to be poisonous. So here is a written description of what I've taught you in the past couple minutes, right, in the diagrams and the drawings. So please read through that yourself. And here is talking about the ATP synthase, which will use the H plus gradient to help create the ATP. So the movement across the H plus across a semi-permeable membrane down their electrochemical gradient is called chemiosmosis. Okay, so by having a high gradient in the intermembrane space and a low gradient and a low concentration in the matrix, that creates an electrochemical gradient. So chemical gradient is just high concentration to low concentration, right? And the electro part is because the molecules are charged, right? So all these H plus are going to have a repulsion force against each other. So combined with the high to low concentration gradient, that's going to cause them to really go through the ATPase right here. So that gradient is called the chemiosmosis. And the ATP produced in the electron transport chain, as we talked about already, uses oxidative phosphorylation. Okay, because the energy that is used to create ATP is actually harnessed by oxidizing NADH and FADH2, meaning the NADH has to give off the electron to the chain. FADH2 has to give off the electron to the chain first, right? And whenever you give off electron, that's called oxidation, right? So ultimately, this phosphorylation, the addition of phosphate to ADP, is powered by oxidation. That's why they're called oxidative phosphorylation, as opposed to substrate level phosphorylation, right? So 90% of the ATP produced in cellular respiration is actually created by this process right here. So now let's try to account for all the ATP that is produced by the cellular respiration. You should know by now that the chemical equation for cellular respiration is 1 glucose plus 6 oxygen will produce 6 carbon dioxide plus 6 water and up to 36 ATP. Okay, I say up to 36 because things can sometimes go wrong, and so under ideal situation, that's how many you can create. All right, so the glucose is going to be oxidized into carbon dioxide. That means it's going to be releasing electrons, and as you strip electrons away from the glucose, they will be picked up by energy carrier, which then will deposit the electron at the electron transport chain, and ultimately the oxygen will be receiving those electrons at the bottom of the ETC, and as they receive the electron, they will be reduced into water molecule. Okay, so the 6 carbon dioxide, right? 6 carbon dioxide, just to help you review. 2 carbon dioxide will be from pyruvate oxidation, which is step 2 of cellular respiration, and then you will have 4 more carbon dioxide created from Krebs cycle. Okay. Now let's try to account for the 36 ATPs that are going to be produced. In step 1, we have glycolysis. Okay, so glycolysis, the key products would be 2 net ATP. Okay, you invested 2, but then you produce 4 in total. So 4 minus 2 gives you 2 net ATP. You are going to be also creating 2 NADH. So normally NADH will be able to produce up to 3 ATP per molecule, right? However, these NADH are actually created in the cytoplasm, because that's where glycolysis takes place. And the electron transport chain is going to be happening in the inner mitochondrial membrane. So these NADH molecules actually cannot go into the matrix by themselves. So instead what happens is they will pass the electron to an FAD molecule that is already in the mitochondria, and so it will create FADH2. Okay, so 2. So in doing so, you will lose a little bit of energy. Okay, so these 2 FADH2 will then deposit the electron to your ETC, and so for each FADH2, you can generate 2 ATP, right? So times 2 ATP per FADH2, so 2 times 2, that's going to be 4 ATP. Okay, so from glycolysis, you will have a total of 2 plus 4, which is going to be 6 ATP so far. Okay, next we have pyruvate oxidation. That is step number 2. Step number 2. Pyruvate oxidation. And again, I encourage you to be doing this with me, so it will make more sense if we do it together. In pyruvate oxidation, we are not generating any ATP. Well, I should actually go back here and mention that these two ATPs that were generated in glycolysis, that was by substrate level phosphorylation. Okay, so no ATP creation in pyruvate oxidation, but we are going to be releasing some electrons, and those will be captured by NADH. Okay, NADH. So a total of 2 NADH per glucose molecule. This actually happens in the matrix. So the NADH will be able to transfer the electron directly to the electron transport chain, and so we will times 3 ATP per NADH. Okay, so 3 times 2 will give you 6 ATP. So a total of 6 ATP from pyruvate oxidation. Okay. Next we have the Krebs cycle. Okay, Krebs cycle. And you can actually pause the video and try to do this part yourself. Okay, go back to the slide about Krebs cycle and try to account for the remaining ATP as well as energy carrier, and then you can resume the video to see if you're correct. Seriously, try to pause the video and do it yourself. Okay, Krebs cycle. We would have 2 ATP. Right, these are going to be created directly through substrate level phosphorylation. And for each glucose, we will have 6 NADH and 2 FADH2. And again, this is going to be happening in the matrix, so they will have no problem taking it to the ETC directly. So for the 6 NADH, we will times 3 ATP per NADH. That will create 18 ATP, right? And for the FADH2, they will be depositing the electron at a lower point on the ETC, so only 2 ATP per FADH2. And that's going to be 2 times 2, which is 4 ATP. Okay? And so, we can, oops, I forgot to highlight this one. So now we can add it all together. Okay, so we have 2 plus 2, which is 4 ATP by substrate level phosphorylation, and then here we are going to have 4 plus 6, which is going to be 10, and then 10 plus 18 is going to be 28, and then 28 plus 4 is going to be 32 ATP. Okay, so 32 ATP by oxidative phosphorylation, right? Oxidative phosphorylation. I'm just gonna put OP for short. Okay, it's not really the official short form, but I'm out of space here. So oxidative phosphorylation, right? These are created by harnessing the energy from the proton gradient, from the H plus gradient that we've generated, and then as they go back into the ATPase, right? Goes through the ATPase, you're going to be able to harness that energy to produce ATP. So together, 32 plus 4, that's going to be the 36 ATP that you are going to be creating for every glucose that you metabolize. As you can see, right, 4 out of 36, that is roughly 10% of all the ATP by substrate level phosphorylation. Not a lot, but I guess it's still significant. And then 32 out of 36, that's roughly 90%. Okay, 90% of all ATPs are going to be produced by oxidative phosphorylation. Okay, so here is a summary that accounts for all the ATPs that are produced in cellular respiration. So here is the same chart, but a little bit more organized in case you need to have a second look. There is a similar chart in the workbook, in the study guide. You can definitely do that once yourself. Cellular respiration is actually quite versatile in that it can use other molecules besides glucose for energy. It's not just glucose. You can use other type of carbohydrates. You can even use fats and proteins. These molecules are actually going to be converted into glucose in some cases, or they're going to be converted in some intermediate molecules that are going to feed into different parts of cellular respiration. So, for example, fats can be broken down into glycerol and fatty acids, right? You know, the fat molecule, right, has one glycerol and 3 fatty acids. So the glycerol can be fed into glycolysis directly, whereas the fatty acids, they're going to be fed into the acetyl-CoA. Okay, proteins, depending on which amino acid it is, you have 20 different amino acids. Some can be fed into glycolysis, others it's going to be converted into acetyl-CoA and go into Krebs cycle, and still other amino acids can feed in the middle of the Krebs cycle. Okay, and so in that respect, it's quite versatile. We can use other food sources as energy. So now we are going to look at anaerobic respiration. Earlier we talked about how the at the end of glycolysis, after you get the pyruvate molecules, then the fate of the pyruvate actually depends on how much oxygen you have, right? And it also depends on the kind of organisms that you are. So if there is sufficient amount of oxygen, then it went through all the stuff that we talked about. However, when we don't have enough oxygen, then we will go into anaerobic respiration. Okay, so in humans we do anaerobic respiration in the form of lactic acid fermentation. In something like yeast, which we use to make wine, as well as a leavener for bread baking, they use a type of anaerobic respiration called alcoholic fermentation. Okay, so let's take a look at how those processes are similar and how they are different. So fermentation in human muscle cells. If you are doing strenuous exercise, okay, you're running really hard and for quite some time now, then what happens is it's possible that your oxygen supply in the muscle is not going to be met by the oxygen demand. Okay, so your muscle cell needs more oxygen than you can supply. In that case, the pyruvate will not be able to go through the Krebs cycle and the ETC, because without adequate supply of oxygen, we don't have enough final electron acceptor, right? So we cannot pull the electrons away from the ETC, and it's going to get clogged. Okay, so as an alternative pathway, the pyruvate will undergo lactic acid fermentation. So in lactic acid fermentation, the pyruvate is going to be converted into lactate, and the purpose of that is so that the NADH can have somewhere to deposit the electron. Okay, remember, when you don't have enough oxygen supply, the ETC is clogged, so the NADH does not have a place to put those electrons. Okay, and so by giving the electron to pyruvate, you will be oxidizing pyruvate into lactate, right, or lactic acid, and that's going to allow you to regenerate NAD+. Okay, and regeneration of NAD+ is important because you will be able to go back to glycolysis and allow glycolysis to keep on happening. Right, so glycolysis can generate 2 ATP. Right, it's not a lot, but when your oxygen supply is really low, that 2 ATP can make a little bit of difference. Right, so that's why the lactic acid fermentation is necessary. Okay, it allows you to regenerate NAD+. So the byproduct is lactate, and the buildup of lactate in the muscle. It's sometimes what gives you like the muscle cramp and the soreness that you have the following day after strenuous exercise. So here you can see glycolysis involves releasing electrons. Those electrons are going to be picked up by NAD+, converting into NADH. Normally speaking, this would be able to go to the Krebs cycle, right? The ETC rather, to generate ATP. But the ETC is clogged when you don't have enough oxygen. So as an alternative pathway, the electron will now go to the pyruvic acid, the pyruvate. Okay, and that will convert the pyruvate into lactate or lactic acid, and you will regenerate NAD+. So that NAD+ will allow glycolysis to keep on happening, so you will still be able to produce 2 ATP even though your oxygen supply is low. So in some microorganisms, they also perform fermentation. They don't have the sophisticated proteins and enzymes and structures to carry out aerobic respiration. Okay, the Krebs cycles and the ETCs, they can't do that, right? So instead, they rely on different types of fermentation. And for yeast, they are going to be doing what's called alcoholic fermentation, which produces carbon dioxide as well as ethanol, which is drinking alcohol. There are different types of alcohol, right? Not all alcohols can be consumed, right? You have like rubbing alcohol, which is isopropanol. You can't drink that stuff. Okay. You have methanol, right? You also cannot drink that stuff. Okay. So the only alcohol that's consumable without being toxic to the body is ethanol. Okay. So because it generates carbon dioxide and ethanol, the food industry, of course, use yeast to produce various food products, including wine and bread. So here, that's what yeast looks like. In fact, this one is kind of budding. It's dividing. And in alcoholic fermentation, after glycolysis, the pyruvate will then be converted into ethanol and carbon dioxide. Instead of in humans, we would have the pyruvate being converted into lactic acid, right? Humans don't do alcoholic fermentation, right? Because otherwise we'd be kind of like drunk all the time with all these ethanol that's produced in the body. It's actually toxic to the body, right? If we constantly produce ethanol in the body, so we create lactate or lactic acid instead. Before these yeast, they're okay with it. And again, the purpose of fermentation is to regenerate NAD+ so that glycolysis can continue to occur. So here is a similar diagram as the one we saw earlier, but now in this case the pyruvate will be releasing carbon dioxide and in the process generating ethyl alcohol or ethanol. So the three fates of pyruvate produced by glycolysis, right? If you have adequate oxygen supply and you are eukaryotic cells, right, with all the machinery required for the next few steps, then you will be doing Krebs cycles as well as oxidative phosphorylation. Okay. If there is not enough oxygen, or if you don't have the machinery to do regular cellular respiration, then you will be doing anaerobic respiration. Okay, so there are two forms. In humans, we do lactic acid fermentation, creating lactate, which builds up in the muscle. For some unicellular organisms like yeast, they will be doing alcoholic fermentation, where they will release carbon dioxide as well as producing ethanol. Okay, but the purpose of the anaerobic fermentation is always to regenerate NAD+, right, so that the NAD+ can allow glycolysis to keep going. So you will at least be getting to ATP instead of none. So that's it for this lecture. Just try to go through the study guides questions that will help you focus on what's important and what's not, and we'll talk more about this during the tutorial sessions. Thanks for watching.